300 million people, 300 million people worldwide are suffering from type 1 diabetes. As we know, this is caused by the autoimmune destruction of insulin producing pancreatic cells. This is something that cannot be prevented, it is genetic, and so far, up until recently, we did not have many treatments for it. There were transplants from cadavers, but they only lasted a very short time. And while also being incredibly expensive, they were hard to come by. There's also insulin treatments, but unfortunately, this is limited by the amount of insulin producing cells that the person has left and how well they are still functioning. My name is Liliana Rodriguez, and today I will be presenting to you about this groundbreaking research led by Doug Melton. Yes, Doug Melton, that has provided us, hopefully, with a pathway to a cure. Now, some previous studies that have been done using embryonic cells have produced something that is very close to what an insulin producing pancreatic cell would look like, which is a beta cell. But they have not gotten it to where it needs to be. They have many groups of researchers actually had been working together to try to get it to the right spot, but they were missing a few things along the way. As you can see in this chart, the group members that were working on this research have only gotten up to this point. They hadn't gotten the cells to produce insulin correctly. The ones that they did have were they failed to produce insulin. They expressed the wrong markers or didn't express them at all, or they had other abnormalities in them, which would not be it would not be useful in transplanting them into humans. What this new group of researchers have done is find the last few things to make this beta cell functional. And so how did they do that? They used embryonic stem cells and incubated them while adding different things into them using this previous research that had already been conducted, and differentiating the cells and getting them closer and closer to where they needed to be. And this, they know that it's going to the right place by the markers that the cell is expressing. And it's not something that is necessary for the cell to have to function, but it's something that we need to see to identify the cell as a beta cell. The next thing they did once they identified this beta cell was that they used an ELISA test to see how much insulin these cells were producing. After they found that the cells were producing insulin, they took it to the next level and transplanted these cells into mice. A mere 2 weeks after transplantation, they again did these tests on the level of insulin being produced by these cells and found that they were very extremely close to the working cells of a regular pancreas. And so on this next slide here, we can see the findings. And on the top row here, you see the different stages of differentiation and this end stage, the beta cell, which this research group got to. And underneath here, you see the, the, the graph of the resulting ELISA test showing the different levels of insulin after 30 minutes of glucose in the bloodstream, or in the cells, I should say. So as you can see here, this is the amount of insulin produced on this axis, and this is the amount of glucose given on this other axis. And they had two different control groups. One of them was of the human cells that we already know work, and the other was from the previous differentiation of cells, which we know didn't work. This was the work done by the previous researchers that couldn't get it to function correctly. And the one that we see here is the test cells, which this group has produced. And as we can see, the pattern, the test beta cells, are reacting extremely similarly to the human cells that work. And the reason that they did these different amounts of glucose was to see how reactive the cells were to glucose. And this was to make sure that the cells wouldn't just turn on indefinitely once they sense glucose, and wouldn't turn off indefinitely when they didn't sense the glucose. This is why they differentiated the amount of glucose that they were testing it with. Now on this other chart, after they found how reactive they were to the insulin, these in vitro cells, they injected these cells into diabetic mice. And after 2 weeks, they did the same thing, the testing of the glucose versus the insulin. And we find that they have very similar results to the previous. The cells are reacting in a pattern. So the control that has differentiation, which we know don't work, are not working. So that's good. Well, not good, but good. The control of the human working pancreatic cells are on point. And the test cells, which we didn't know if they were going to work or not, they are following a very similar pattern to the human ones. And this is groundbreaking because the previous working cells that were produced took 4 months to differentiate enough to be functional. And this is a very long time. And unfortunately, they were not able to directly see what was happening inside of the mouse and see what was happening that changed them into functioning cells. So that was something that wasn't going to work. Some implications about this research is that it paves the way into finding the cure for diabetes. And we would do this obviously by doing clinical trials in the future. But a problem with this sort of research.
Please, the time is now 11:30. The library will be closing in 30 minutes. Restrooms, the study room, the elevator, and the entrance to the work level will be closed in 15 minutes. Laptop computers must be returned in 15 minutes. The library opens tomorrow at 7:45.
Okay. Anyways. Anyways, one of the implications that we have with this type of research is that it's using embryonic stem cells, which are harvested from human embryos. Now, this is an ethical problem that is hard to solve. Well, it's hard to really pick a side. Do we use stem cells from human embryos to do this research, or do we not use them and not get the results? It's something that everyone has to choose for themselves. But if we do continue to do this research, there is much hope that we can find a cure sometime